So in the interest of time, I think we are starting the session. So I'm very glad to introduce this session in pediatric extrapolation, where we will have also a couple of examples of where it has succeeded or, not failed, but yeah, maybe it didn't work as expected. And the first speaker today is Dr. Christine Carlson. She has been employed as a pharmacometrician and pharmacokinetics senior assessor at the Swedish Medical Products Agency since 2016. In her role as senior assessor, she has been involved in assessing questions related to modeling and simulation in various procedures, such as initial marketing authorization applications, new indications in pediatrics, and scientific advice. She is the vice chair of the newly formed EMA Methodology Working Party and was the chair of the former Modeling and Simulation Working Party till 2022. And she also is regulatory chair of the ICH M15 Informal Working Group, and previous member of the ICH E11A Expert Working Group, Pediatric Extrapolation. Christine has a chemical engineer, is a chemical engineer, and earned her PhD in pharmacometrics at Uppsala University. And Christine, she's going to talk to us about pediatric extrapolation opportunities and challenges. So the floor is yours.
Thank you very much, Rocio, and thank you, PAGE Organizing Committee, for inviting me to give you an introduction to pediatric extrapolation from a regulatory point of view, which I call opportunities and challenges. And first, the mandatory disclaimer that what I present here today is not necessarily the view of MPA, Swedish Medical Products Agency, or EMA. It's my own views. So I would like to start with a little bit of history of pediatric extrapolation in EU mainly, to set the scene a little bit. Why is this an increasing talk about extrapolation and the hype about it? So in 2007, there was the start of the EU pediatric regulation, and since then there are, with some exceptions, mandatory pediatric investigation plans in most drug development programs. So that meant that it needed to be a bit more carefully thought about how to develop drugs for pediatric population. And then it took some time, but in 2017, there was a revision of the ICH E11 guideline. It's a guideline on clinical investigation of medicinal products for the pediatric population. And when that revision took place, it was identified that there is a need for a guidance on pediatric extrapolation specifically. So that initiated the start of the work of that guideline. And almost at the same time, EMA came out with an extrapolation reflection paper. And in that reflection paper, it's highlighted that the role of quantitative approaches is very important and can deliver key evidence in an extrapolation setting. And last year, there was the public consultation, so the published draft of this new ICH, which is a global guideline for pediatric extrapolation, and the plan for that to be is to be published or final next year. So that has built up. There's some more buzz around pediatric extrapolation in the regulatory setting, so that's also spills over to all of you in this room. So what is pediatric extrapolation really? So in the ICH E11A new guideline, it says that pediatric extrapolation is defined as an approach to provide providing evidence in support of effective and safe use of drugs in the pediatric population when it can be assumed that the course of the disease and the expected response to medicinal product would be sufficiently similar in the pediatric and reference population. And furthermore, in the EMA reflection paper, it says that it is appropriate to take advantage of existing information when planning and evaluating clinical studies in children. A more targeted generation of evidence should help to ensure that children only participate in clinical trials with specific objectives that further the scientific understanding of a medicinal product for use in children and address the requirements for regulatory decision-making. So both, it says that you should try to integrate as much knowledge as possible, but also the incentive behind is that we want to have good, well-established drugs for children, but we also do not want to put children in clinical trials that are not necessary. So that's where the whole thinking about extrapolation comes from. The extreme would be that we have to do full programs, drug development programs in children all the time, which is of course not a good approach. So where can we leverage information from other populations, either adult populations or other pediatric populations? So along those lines then, in the new ICH guideline, it has been a pediatric extrapolation framework has been put forward. And it has sort of three pillars. You have disease similarity, you have drug pharmacology, and response to treatment. And all those three pillars have to be sufficiently similar, or you have to have some understanding of how those will behave in the pediatric population compared to an adult or a reference population. So then another new term has been put forward, the extrapolation concept. And that is to integrate the evidence and identify knowledge gaps in these three pillars. So to know to what extent you can extrapolate from another population to the pediatric population, you need to compile all the evidence that are out there to have a good foundation for your way forward. And that's, as we pharmacometricians know, that modeling can be a very good way of integrating knowledge. This is a very good approach forward for us. But it could be that you need to have additional data collection. You see that you just don't know. So that could be in adults. We need to have more information for a certain biomarker or for something in adults to be able to know whether we can extrapolate to children. Or you can compile a nice justification for your extrapolation plan, which is then what you need to study in your pediatric population. And this is, of course, a sort of an iterative process, or could be. Hopefully it's not. Hopefully it's linear, and you can get to your dosing recommendations in children once the pediatric studies are performed. And going into the extrapolation concept a little bit more. So this is then, as I said, how you justify what needs to be studied. And it's again the similarity of disease, it's the response to treatment. So you can have we know that disease is very similar in adults and children, but for some reason, we are not certain about whether the response to this treatment will be the same. So then you need to sort of map out what information you have about this. Where are you in this very red part of the arrow, and then you need to study more in children, or very green, and you can extrapolate more. And so that's the large data gaps, is the red part, and the high quality data or high confidence data. And that would be, for example, in an antibiotic drug development setting, we know that the bacteria will behave the same in an adult population and a pediatric population. So the only thing we need to know really is that the dose is right in children and that the exposure will be correct. And to be able to compile this information, and of course, if you're at the extremes, that's sort of the easy part. You know that if it's dark red, you need to do full programs in children, or you say that this is just not possible. Or if it's very green, you can extrapolate a lot. If you're really, really lucky or in rare circumstances, you can just generalize the dose from adults to pediatric population. But it's in the middle. That's the hard part, to know how do we really assess whether its disease is similar, whether the response to treatment is similar, and the drug pharmacology will behave in at least a predictable way. It doesn't have to be similar necessarily, but we need to understand how it will behave in children. So when we have done that, and we now know what we need to study in children, how do we do that? Oh, sorry, I forgot my bullet. Quantitative methods can be used to synthesize and integrating existing evidence. So this is where our role comes in, to really try to piece all these pieces together. It could be in one model. Maybe that will be very difficult, but we could have several models that piece this information together. And when we have done that, we'll go over to the pediatric extrapolation plan. And that's, again, the studies that you will perform in the pediatric setting. So the very red one, again, you need to study, have an efficacy trial in children. The very green one, you only need a PK trial in children to get the dosing recommendation. But the hard part is the sort of gray zone in the middle, where you can think of, you can bridge biomarkers. Are there other response markers that can be used? There's also an increased appetite for Bayesian strategies or modified frequentist approaches, where you use some prior information from other reference populations. Or you can also think about single-arm PK/PD studies. There is a wealth of opportunities in this middle area, but also it's also challenging to understand what you can do and what the information that comes out of it. And modeling here can be really, really important to examine appropriate study designs for these trials, to inform on the dose selection, what doses to study in the pediatric trials, and also perform sensitivity analyses, because there's going to be a lot of assumptions. Some are much more easy to interpret than others. And if they're difficult to interpret, it might be very important and good for us regulators to perform some sensitivity analyses on what would happen in a sort of worst case scenarios, or if it would look different than we thought. So there are some challenges, as I've already alluded to, in this extrapolation concept plan, extrapolation framework setting. The number one that always comes up, how similar is similar? And it's it's a case-by-case situation, I would say. It's very difficult to know what's similar, and that's why we need the whole extrapolation concept. We need to compile the information that we have in adults. We might have some information in other indications in pediatric setting to understand how similar do we need to be. What does what's the side effects looks like? What that can be problematic. But it is a really difficult question to answer. And I know everyone would like to have similar is the number 5. And I don't think that's going to ever happen. It is a puzzle to piece together, but we can all be a bit clearer and more structured than in the past in how we assess and how we tell or justify how similar it needs to be. Another challenge from a more modeling point of view is to
1 minute left, Christine.
Thank you. Provide sound quantitative evidence to support disease similarity and response to treatment in small populations. Because, of course, if we have small populations, the models are also going to be uncertain because there's just little data available. Models in early drug development often come with high uncertainty, which also is a challenge for us. Design of informative clinical trials in situations with feasibility and ethical constraints. It's not only the science, it's the feasibility and the ethics of studying in children that provides challenges. And extrapolation, I just want to emphasize that it does not equal collect less data or just smaller trials. It should be informative data. And opportunities, model-informed approaches to justify the extrapolation concept. I've gone through that already. We also have a great opportunity to inform study size. It's always a big issue. Dose selection, duration of treatment, and define success criteria. What do we need to, how do we need to match, or how do we show that this is similar to the adult population which we extrapolate efficacy or safety from. And model-informed approaches or analyses often essential in support of drug dosing and recommendations in pediatric patients. So exposure matching, it's always model-based, and you can also use simulation-based dosing recommendations in certain circumstances. So I just want to end on a good note, although there are a lot of challenges, that model-informed drug development is recognized as an essential part of pediatric drug development, and in particular to support extrapolation approaches. Thank you for your attention.
Thank you, Christine. So hold your burning questions and comments for later in the session, because we plan to have a bit of a Q&A with Christine and the audience at the end, if there is time, hopefully. So in the interest of time, then we move to our next speaker. He's Dr. Italo Pogliesi. He's currently Senior Director in Clinical Pharmacology Life Cycle Management at GSK. He holds a degree in chemistry and PhD in bioengineering at the University of Pavia. He has held different positions in the pharmaceutical industry, including Pharmacia, GSK, and J&J, and more recently as consultant at Certara. His research interests include the development and evaluation of modeling techniques to accelerate the development of new drugs, especially in the area of oncology. And today he's going to present us an example of extrapolation in pediatrics, so the extrapolating treatment to children less than 6 years old, the successful story of Belmanas alpha and alpha mannosidosis. So the floor is yours.
Thank you. I would like to thank the organizing committee for giving me the opportunity to present this work of myself and my partner in crime on this interesting program. This is just a brief summary. We'll go through the alpha-mannosidosis as a disease and Lancide as a drug that has been used and has been approved. And we will go through the modeling and extrapolation to pediatric subjects. And here the exercise was to extrapolate in a population of less than 6 years of age. And there are very interesting aspects overall in this exercise. And the first thing is the definition of what is an ultra-rare disease. Here in the schematics up here is the definition of European for rare disease. Rare disease is something that has a prevalence of 1 over a few thousand. Ultra-rare disease is less than 1 over 50,000. And for alpha-mannosidosis, we are in really the extreme cases. We are talking about 1 over 1 million. And the database that is available for patients with this disease is counting approximately 200 subjects overall, which is a very, very small number. And naturally, this requires some eye of regard. The alpha-mannosidosis is a genetic disease, is a progressive genetic disease caused by a mutation in a gene, which is essentially encoding for the metabolism of oligosaccharides. It is involved in one of these steps, and naturally the enzyme is dysfunctional and is leading to accumulation of oligosaccharides in systemic circulation in the system, in the tissues, and in the excreta. And this results in a multimorbid disease. Essentially, the clinical presentation of this disease can be very, very heterogeneous. These subjects are prone to a lot of infections, and in the vast majority of cases, this results in otitis that is leading to hearing loss or hearing impairment. We have skeletal abnormalities. For instance, there are some typical coarse facial features that are occurring in this subject. There is there are motor problems, and this is the basis of sometimes of the functional tests that are used to as an endpoint, as a clinical endpoint in these cases. There is mental retardation in these subjects. Okay. Now the compound, velmanase alpha, called Lancide, is a recombinant form of the human enzyme. And essentially the compound is identical to the naturally occurring human enzyme. This is an enzyme replacement therapy, so essentially the idea is to provide and substitute the deficient enzyme to subjects that need it. And this compound, as well as the natural form, is eliminated via the normal processes of protein catabolism. The compound is currently approved in both EU and US for the treatment and for the treatment of the manifestation of alpha-mannosidosis. And essentially at the bottom here you can see the objective of this exercise. We had to update a pre-existing population PK model. We had to develop the exposure-response relationship with an endpoint of pharmacological activity, that is the serum oligosaccharides concentration. And in particular, we will describe the extrapolation to support the use of velmanase alpha in pediatric population without age limitations. So here is just a schematic of the data that were available. We are talking about 10 studies, starting from a dose escalation study. The compound is given IV, typically as a 1 or 2 hour infusion, and it is given weekly. So following the dose escalation, the phase 2 trials focused the attention on a more limited range of doses. At the end, the clinical dose was decided to be 1 milligram per kilogram, so a dose based on weight. The important thing is that we had a phase 3 trial, which was a comparatory study with placebo, in a population of subjects between 6 and 35 years, so spanning from some of the age range for pediatrics and adult subjects. At the very end, there was a small studies giving a look to PK and oligosaccharide concentration in subjects lower than 6 years. The interesting aspect is that due to the ultra-rarity of this disease, many of the subjects were rolled over from one study to the other. Naturally, the phase 3 trial was in naive subjects, and also the smaller kids study was in naive subjects. In addition to this study, there was a natural history study which indicated a very strong correlation of the oligosaccharides concentration in both urine or serum with endpoints of clinical status. So that this indicated that this marker could be used as a confirmatory evidence. Could not be used as a surrogate marker, but at least as confirmatory evidence. This is essentially the schematics related to the approach we use for modeling and simulation. We had the pharmacokinetic model redevelopment and the model development for serum oligosaccharides. And then what we did to extrapolate was to simulate the pharmacokinetics in a virtual pediatric population and use the exposure response model for simulating the serum oligosaccharides in virtual pediatric populations. Essentially, the idea here was that the phase 3 trial was going down to 6 years of age. There were no formal confirmatory data on clinical benefit on subject below that age, so that the idea was to support that with an extrapolation exercise. Here you can see some of the characteristics of the populations that were included in this exercise. We have a small number of children lower than 6 years. We had overall 27 subjects in a pediatric range and 12 in the adults. In this exercise, the idea is that the compound is a large molecule, naturally is an enzyme, so there is the potential for developing anti-drug antibody, and in this redevelopment of the PK model that was considered. And here you can see some of the information related to the occurrence of ADA in this population. Quite interestingly, there were essentially no subject developing ADA in the adult populations, whilst they were present in the pediatric one. And the other levels were very variable across individuals. So here is the first bit of the modeling exercise, the population PK. This was, we used a 3-compartmental model with constant rate infusion. The body weight was accounted for using standard allometric coefficients, and we incorporated covariates. We examined covariates in particular. We had an eye of regard for the ADA presence. Those levels for giving a look to the potential dose nonlinearity. And there was also something to be accounted for in terms of inter-study difference. Naturally, age and sex were also explored. Overall, the model was doing its job. It was a relatively simple model. The model was able to describe the observations both in subjects without development of ADA and in the subject with the developing ADA. The pharmacokinetic parameters were essentially in the range expected for this sort of compound, so pretty slow clearance, pretty minimal volume of distribution. Here are the oligosaccharides concentration. On the left-hand side, the overall data. In the black dots are the oligosaccharides observed in placebo-treated in the phase 3 trial. Overall, it can be seen that there is a quite good inhibition of these concentration in subject treated with this compound. And on the right-hand side, you can see let's say the average data for all the subjects involved in this exercise. This is the model for exposure response. It is a simple inhibitory Emax model. The baseline was essentially in the range expected of 7 micromolar. We had a maximal effect of 77%, and we have an EC50 which is pretty low, meaning that essentially all subjects, including those that were in whom the PK is influenced by the other development, were well exposed in order to have the effect on this biomarker. Giving a look to the 3 pillars of extrapolation, so the similarity in disease progression between the general and the extrapolated population, the similarity of pharmacological characteristics, and the similarity of the response. Essentially, alpha-mannosidosis is an inherited progressive disorder, so it is sensible to consider that this is a continuum between subjects lower than 6 years and above 6 years. The pharmacological also is relatively sensible to consider that this is an enzyme replacement therapy, so the pharmacological basis of the action is there. And we had also that the pharmacokinetics was independent of age when body weight was considered. And in terms of the similarity of the response, the very limited data we had in subject in the smaller kids indicated that the exposure response was independent of age. So what we did was to mine the NANES database to generate vehicle
Can you
population.
try to resume? Sorry, because we are out of time. So.
Okay. Anyway, these are the results. So we have the pharmacokinetic, as can be expected based on the dosing. We have some difference in the different population, but when we were giving a look to the oligosaccharides, essentially all subjects were expected to have exactly the same response. So. I think this is a success story. We have substantial evidence of effectiveness. The safety profile is acceptable for the intended use, and the serum oligosaccharide model-based extrapolation warranted the approval without age-related limitation. Only another minimal consideration is related to, in these ultra-rare cases, regulatory flexibility is warranted. There was no statistical significance based on the phase 3 trial, but this was counterbalanced by the strength and the design of the conduct of the study. So essentially, the confirmatory evidence and the oligosaccharide data were substantial, were substantiating the evidence of effectiveness. Also, something I noticed that the health authorities were asking for a number of additional ER analysis, even if the sample size was very small. I guess that this was due to the intent of build-up knowledge in this area. So apologies for delay, and thank you very much for your attention.
Thank you. So I think we have time for a couple of questions from the audience. And in the meantime, I can ask you, so I'm interested to know, I mean, there is a variety of mutations, I understand, in the disease, and how does that impact on the level? I mean, are there differences in expression of the enzyme across the patients and the variability, and how do you deal with that then in your model? And because it may impact then in the dose, I guess. So if you could comment on that.
That's a very interesting question. I was mentioning that we have a database of approximately 200 patients, and we have among these patients 154 different mutations. And it is reported in the literature that even siblings with the same mutation have different clinical presentations. So that was not really possible to be accounted for, but that's an interesting aspect, and I don't know whether there will be further, let's say, advancement in the knowledge that might help.
Okay, thank you. No questions from the audience? Well, you will have maybe a chance later again. So I think we move to our last speaker in the session then.
Yes, and with that, I would like to introduce Bart van Lieshout, who's a master's student in biopharmaceutical sciences at Leiden University. And presently, he's doing an internship at the Amsterdam University Medical Center, working on a project that he will present for us now, which is titled Population Pharmacokinetic Analysis of guanabans in Children with Vanishing White Matter. Please, Bart.
Yes, thank you for the introduction. So I'm here on behalf of the team behind the clinical trial of guanabans for vanishing white matter, here to share with you the pharmacological approach of the clinical trial and the results so far. Starting with an introduction on the disease, as not everyone might be familiar with it. Vanishing white matter, as the name suggests, is a inherited leukodystrophy that leads to the degeneration of white matter in the brain. This, as you can see from the image of the MRI scan, can lead to severe impact on the patient's cognitive and motor functions, and this impact is even more severe in children, which makes it all the more unfortunate that there's currently no treatment for vanishing white matter. And that highlights the need for research into potential drugs that can serve as a treatment, and is why we are performing our clinical trial. And that brings us to one of those candidates. The main subject of the trial, guanabans, is an antihypertensive agent that has shown to have a beneficial effect on the disease's progression. As you can, in a study with vanishing white matter mouse model, it had a positive effect on the neuroscore, which reflects the brain's deterioration. It is decreased in the group that received guanabans compared to the group that compared to the placebo, and it improved the motor function of the mice in physical experiments. And these promising results are why we have set up a phase 1/2 clinical trial, and it's in a pediatric population, and as the subject of this session is pediatric extrapolation, I would like to share with you how we determined the dose, how this turned out, and the results of the study so far. For the dose determination, we had to start with developing a pediatric model. We did this by taking the data that's available for adults, and this data, the pharmacokinetics of adults was best described by a one-compartment model, and the corresponding PK parameters were then extrapolated to a pediatric model by using allometric extrapolation on clearance and volume. The enzyme maturation was also taken into consideration, but the age of our patient population in the trial is above the age where the primary enzyme from guanabans metabolism is fully mature. For the pharmacodynamic side, we have data on some side effects after 6 months of guanabans dosing in adults. Some of the side effects are insomnia, headaches, and drowsiness. And we also have data on the effect of on the blood pressure, where we can see that the maximum effect on the blood pressure, on diastolic blood pressure, is around a decrease of 20 millimeters of mercury. This data, alongside other adult data and the data of the vanishing white matter mouse models and other rodent neurological disease models, were then used to determine the therapeutic window of guanabans. We performed simulations of several doses with the rodent models, the adult models, and the extrapolated pediatric model to see which doses were best suited for our trial. And we determined that the starting dose would be 0.5 milligrams per kilogram, and we titrated this upwards in the clinic to the maximum individual tolerated dose, or the upper limit of 10 milligrams per kilogram. For the effectiveness, we determined that the optimal concentration, optimal dose would be 2 milligrams per kilogram, but the minimum effective dose would be 1. We dose once daily in the evening to circumvent the significant effect of drowsiness. And for the sampling, we've coincided our plasma sampling with the annual MRI scans of the patients, which is performed under sedation. And we can therefore use the IV line that's set up for administering the anesthesia to take the plasma samples without adding any invasive procedures. Additionally, we've used a less invasive sampling method, which are the dry blood spots, and we take these every 3 months. Before we go ahead into the results, we can go over some of the central questions of the trial. The main question for clinical trials, of course, the pharmacodynamics and the tolerability of Quanabens in children. And these two, by answering these two questions, we can also, as a bonus, evaluate our extrapolation approach. And the first way that we want to evaluate this is by looking at the PK profile, whether the PK profile that is described by our extrapolated model corresponds to the observed concentrations or the observed PK profile of the children. And we evaluate just by looking at the prediction error of the two of the observations and extrapolated predictions and the visual predictive check. And additionally, we also try to improve the extrapolated model by performing re-estimation and stepwise covariate modeling in non-MEM to see what the differences are between the extrapolated model and a model that's tailored to our population. Additionally, we want to see, we want to look at whether we reach the doses that we determined with our with our approach. And we do this by looking at the adverse events and looking at whether there are covariates that are predictive of these events. And we can also use our fitted PK model to look to investigate whether there's a concentration response relationship with these adverse events. And lastly, we want to look at biomarkers for the brain accumulation, because the beneficial effect happens in the brain. We want to be able to somewhat quantify the amount of Quanabens in the brain without measuring it. And because the effect on the blood pressure also occurs due to binding to receptors in the brain, we want to look at whether we can use the decreases in blood pressure as a biomarker. For the first of the results, we have an overview of the characteristics of our population at in the trial so far. For the, we currently have 26 patients that are included in our trial, 10 of which have already had a second set of plasma samples taken. For the current doses, we are in the range of 1 to 1.7 milligrams per kilogram, and that is below the optimal concentration, but at least above the minimum effective concentration or dose that we determined. And when we look at the C trough concentrations after 1 year, we can see that they are about as high as the C max concentration at the baseline dose. And through that, we can determine that the patients are adherent to the Quanabens therapy and that they've reached a steady state at that point. For the model predictions versus the observations, we can see that there is an under-prediction of the observations. It is on the twofold difference mark with an under-prediction of minus 50% defined by the median prediction error. But there's still room for improvement. And we did this by looking at several structural models and performing re-estimation of the parameters and performing stepwise covariate modeling. And the main changes between the final model and the extrapolated model are a decrease in clearance and volume and different allometric scaling exponents. But these changes lead to a model that can quite confidently predict our population's concentrations. For the pharmacodynamics, we have the adverse events. The severe events were quite uncommon, but the less severe events were much more common than we have seen from the adults. But it's to be expected because we dose to the maximum individual tolerated dose. But to alleviate this somewhat, we have decided to split the doses with a smaller dose in the afternoon and a larger dose in the evening. And then we get to the hallucinations. We did not expect these from the adult data, but these are quite common in our population. And it's also one of the reasons that we could not reach the optimal dose. And therefore we wanted to look at the covariates of these hallucinations. And therefore we have looked at the time to event and the Kaplan-Meier analysis on the left. From the time to event plot, you can see that most of the hallucination events occur at the start of the trial during the titration phase. And we also observed that they are very common in the younger children or the youngest children in our trial. And therefore we have split our population in two across the mean age. And as you can see in the Kaplan-Meier plot, where the oldest population has an incidence of about 20%, and the youngest population has an incidence of about 53%. And although this difference is quite large, it's not a significant covariate. So we also looked at the concentration response relationship by using the fitted PK model and simulations of the doses during the titration phase. And here you can see that for the AUC and Cmax, the first few days, the hallucinating group has a higher value than the non-hallucinating group, but this difference quickly levels out. And if we include the concentration response relationship in time to event models or logistic models, there also wasn't a significant effect of Granabens on these hallucinations. Lastly, we have the blood pressure data that we wanted to model to investigate whether it could be a biomarker for the blood concentration. We tried several models and based on the characteristics of the observations of the decrease in blood pressure after a dose. These characteristics were firstly that the time point of the maximum effect was after the time point of the maximum concentration in the blood, and as we increased our dose during the titration phase, the effect of the decrease in the blood pressure didn't become longer or it didn't become greater. So we included an effect compartment for the delay that can resemble the brain, and an exposure component that reduces the effect based on the exposure of the effect compartment. And the predictions made by this model are quite good, but the question isn't whether we can predict the blood pressure, but whether we can use this as a biomarker for the concentration in the brain. And as there's a tolerance component that changes the concentration response relationship, it might be challenging to accurately quantify the true concentration in the effect compartment. And that brings us to the conclusions and discussion. Firstly, for the PK extrapolation, we used the allometric extrapolation, and this led to an under prediction of the pediatric concentrations that we observed, and the allometric scaling worked well, but the exponents that we fitted were higher, so it might not be the best approach. Then for the doses that we reached, or the dose that we determined, we didn't reach them, mainly because they had hallucinations, but those weren't to be expected from the data that we had. And we didn't find a significant covariate or concentration response relationship, but we're still conducting the trial, so moving forward we would like to have some idea on how to prevent these hallucinations. So it would be best to either more extensively monitor the youngest patients during the titration phase or titrate slower. And lastly, for the the blood pressure biomarker, we saw that the tolerance quickly develops, which is great for the tolerability and safety, but it does obfuscate the actual concentration or a direct quantification between the blood pressure and the concentration in the brain. But in the end, when we eventually get the effectiveness data on eventualized matter, we could still take a look at whether there's a better relationship between the plasma concentrations or the predicted concentrations in the effect compartment. And that brings me to the end of the presentation, and I would like to thank the team behind the clinical trial for their hard and diligent work of setting it up and conducting it, and for bringing the possibility of a treatment for Ghana events, for eventualized matter by Ghana events close to reality. And I would like to thank them again for the opportunity, for giving me the opportunity to work on the project. And I would like to thank you all for listening. And are there any questions?
Thank you, Bart, for a very nice and timely presentation. Do we have any questions for Bart?
So, thank you very much. I'm Siv Jönsson from Uppsala University and Prometheus. So, as I understood, you re-estimated the PK model, and
Sorry, go ahead.
including the children data and the allometric exponents. They were not according to those theoretical ones. And is that expected, and why did you re-estimate them? Was it not possible to get a good fit without re-estimation?
Well, we mainly wanted to re-estimate it by getting the most accurate model in order to, one, see the differences between the theoretical extrapolated model and a model that's the best fit to our population, but also primarily because we wanted to be able to perform simulations for discovering concentration response relationships, because we only have data on the very first day, but we do have data on the doses that were given and when, so we were able to simulate the concentrations, and we want to do it as accurately as possible, so we re-estimated the model.
Okay, so if I just rephrase my question, so it is a small molecule, right, this drug?
Yes.
Yeah, so then it's sort of expected that they should use the theoretical ones, but you needed to re-estimate them to get the model to fit the data, right?
Yeah.
Okay, thank you.
So I can kind of follow up on this. Thank you, Siv, for raising the issue. But the problem is with theoretical allometry, it's got a theory behind it, and it says that what you can explain is based upon body mass. And that's the theory. And if you change the exponents in order to fit the data better, which is what you did, you need to think about, as I think Siv asked you, why did that happen? And there are some answers to the questions why. Is because there are other factors that you've not recognized yet. For instance, body composition is one of the factors. If there was a younger age group, not relevant here, but a younger age group where you hadn't accounted for maturation of enzyme function, then those are factors that could cause to incorrect estimates of these exponents. You estimate values that improve the fit, but they actually haven't challenged the theory-based exponents. They're just empirical fudge factors, which shouldn't be relied upon for extrapolation. And this session's about extrapolation. So I understand what you did, but I wouldn't approve, I'm not a regulator, but I wouldn't approve of what you did, of fudging the factors. I think you needed to do a better job of looking for other factors that might have explained why the clearance was underpredicted.
Yes. Thank you. I get your opinion. There are definitely different approaches that can be taken. We have just used the allometric scaling of weight on volume and clearance, but you can also take a PBPK model or using different exponents based on age groups. But in the end, we took the approach of using allometric scaling, and then we showed that it leads to an underprediction. So I wouldn't say it's the best approach. We just highlight that if you use this approach, you might be this far off, so an underprediction. And there's definitely a need for different models.
We have a question in the room.
Closer to the mic? Does it work?
No.
No? Should I talk really loud? I have quite a loud voice.
You could try.
Oh, maybe I can try this. How about this?
Yes, much better.
So I had a question not on allometric scaling. I was wondering for the hallucinations, the plot you made on the time to event sort of looked like a single time to event. But I imagine that this occurs more frequently than once and it's not terminal.
Well, actually, it really only occurs once. If I recall correctly, there's only one patient that had a second event, but for now I just used a single time to event because most of the cases were only a single event. And these were concentrated near the titration phase. So at least we know that if we could take more care during the titration phase, the chance of for the hallucination occurring after it might be very low because it only occurs mostly once and during very start of the trial.
Yeah, so did I hear correctly that you did an up titration to test if that worked? Or?
Yes, we started at 0.5 and then titrated upwards to the maximum in tolerated dose every 3 days.
And that was also done in the ones that developed the hallucination?
Sorry?
That was also done in the ones that developed the hallucination?
Yeah, I mean, it was done in every patient. And depending on the side effects or adverse events that we occurred, we either temporarily decreased the dose or halted the dose, increase for a while until they had time to adjust. But in the end, all the dose adjustments that were able to be made did lead to a dose above 1 milligram per kilogram that we determined would be the minimum effective dose.
Okay, so the hallucination was really a temporary hurdle you could overcome by lowering the dose and then going back?
Yeah.
So would you then recommend the general dosing schedule to be a titration? So to sort of, sometimes you have a side effect that only occurs when you peak immediately, and if you slowly up titrate, it doesn't occur. So then you can give the drug to all patients, and they can be free of hallucinations, maybe.
Yeah, but all patients are titrated upwards.
Yeah, but there could be a slower titration.
Yeah, for sure.
Yeah, thanks.
Sorry, my chair just broke. So, well, thank you, Bart.
Oh.
And I think, well, an applause. And although we are a bit delayed, but we started 3 minutes late, so I think it's worth to open questions also to give the chance to the audience for Christine. On general questions on pediatric extrapolation and examples, if you have any questions from the audience. Or from the
I have
chair.
a comment, which is really should have gone to Italo earlier, where he concluded that something about they had an age-independent dosing schedule because they'd account for weight. But what he missed, and I say Bart didn't miss it, what he missed was the possibility that there is maturation of clearance at ages younger than the lowest age, I think it was 3 years. Maturation is usually happening under the age of 2 years. So I think that was, for extrapolation to younger age groups, that model was incomplete because it didn't have the maturation. And just to say for extrapolation, just to knock on the extrapolation thing, the extrapolation model for maturation should be plausible. It should extrapolate to 0 at 0 age, and it should extrapolate to the adult value as the age approaches adult values. So that's just a comment in general about this session, which is about extrapolation, which we didn't hear much about in the two presentations.
Thank you, Nick. So we have a question here, or a comment, yeah?
Yes, so Stefan Schmidt from the University of Florida. I have a general question regarding the establishment, and I think you call it an extrapolation framework, which I scientifically agree with. What does it mean for the individual sponsor? Because, I mean, usually if you establish a framework, you need to inform it with multiple drugs. So what would happen if you were, let's say, the first player in a certain space? Could you then still use that framework concept? How extensive does it have to be? And what does it mean if you're like the 5th or the 10th in the space? Does it mean that you need to use like data from all your competitors and then basically subsequently give competitors a leg up? So how does that work?
So there we go. The intention with the framework is to be for a single development program. So it's not, it doesn't have to be a disease platform or a multiple compounds. But of course, if you're 5th in row, you have more information available to you. But the framework can be used when you're first in class, and then you have to compile the information that is available to you and to provide sort of a structured justification for why you think it's reasonable to do any extrapolation in this particular setting. So it's more of a, it's not a full, it's not a framework outside your sort of own program. That's not the intention with the guideline, but the more information that's out there, the bigger you can build it. And of course, if a consortium or some people want to go together and provide a sort of disease similarity model for a particular disease area that can be used by multiple sponsors, that's of course a possibility.
Yeah. The second question I had was, does this framework hold both for small and large molecules or novel entities? And can it also be applied to switch between, let's say, immediate and modified release formulations?
So for the first, it's definitely both small and large molecules. For this particular framework, as I showed it here, it comes from the Pediatric Extrapolation Guidelines, so it's particular to pediatric extrapolation. With that said, the similar it's similar approaches when you go from immediate release to modified release. So of course a similar approach could be taken.
Another question here?
Yes, Alan Farai, Uppsala University. So as a PhD student, it's nice to take a step back sometimes from the code and to look at strategy and in particular extrapolation of pediatrics and pediatric clinical development programs. It's actually a question and a comment. I was thinking about the second presentation, and maybe you can, Christine, give some comments on that as well. When I listened, it appeared that there was efficacy data available down to 6. And I'm wondering, how would the extra trial that you did, the phase 2 trial that you did in the end, below 6 years of age, how would the design of that change if you only had efficacy data down to 12 or down to 18? Was the prior trials designed with the intention to go down to 6 so that you do not need to do an extensive trial below 6? Was that the intention from the start, or?
Yeah, you can comment, Italo, please.
I guess that the aspect there was to complement a phase 3 trial that was done in a more feasible population and try to extend the conclusion to the smaller kids. I hope that can be.
Because the extra phase, The extra phase 2 trial that you did, you only looked at PK.
It was only PK and oligosaccharides.
Oligosaccharides, okay. All right, thanks.
You're welcome.
If I could just follow up in terms of the extrapolation of what was called efficacy, but the effectiveness of this drug was to try and maintain white matter in the brain. So presumably at birth, I don't know, but at birth presumably the white matter is more or less normal. It may not be, but presumably the white matter gets worse with time. So the extrapolation problem is to extrapolate from adults with the disease up to newborn babies who have not yet manifested the disease. And that's a very hard extrapolation problem, except you do have a point where you can extrapolate to the baby. But how you link the points is hard. But extrapolating drug effects is typically hard, except in a case like this, where you might actually know what the effect really is in a baby, how much white matter is in the brain.
Okay, so I think, well, with this final note from Nick, we can close the session because we are, overdue and everyone wants to go for lunch. So I invite you to come back at 2 p.m. For the tutorial on machine learning and pharmacometrics. So thank you, everyone, and thank you to our presenters.